Hello, can everybody hear me?
Yes.
Yes.
Okay.
Yes, and on your videos.
Hi, everyone. Thank you all for being here today. I'm Jeffrey, and this is our group's presentation on our project, Elements of AI in Optimization of SEM.
Okay.
Before we start, we have two advisors who helped us through this project, Dr. Starstina and Mr. Downing, and I'd like to thank them for all they've done for us in these past few weeks.
No, thank you. Seriously, thank you guys.
To give a brief introduction on our topic, I'll be talking about material science, electron microscopy, and scanning electron microscopy. So first of all, material science is a field, it's a field of physics that studies the relationship between a material's structure and its physical properties and the behaviors of different substances. It's used to understand why materials have certain physical properties, and it's all about finding out what certain substances are made of, how they're structured on the smallest levels, and how its composition affects its properties. Within the subject of material science, there is a special type of microscopy called electron microscopy, or EM for short. And electron microscopy is a material science imaging technique which uses a beam of electrons to visualize surface and internal structures of small samples which are too small for regular optical microscopes. To put this into perspective, the sample sizes for this microscope need to be within 10 centimeters by 4 centimeters, and the magnification of this microscope ranges from 20 times to 1,000,000 times. To go deeper, there's another type of microscope called a scanning electron microscope, which is a type of electron microscope, but it produces a high resolution and high depth of field image of a sample's structure and
Have you saved the frame there?
the resolution for this.
Yeah, once. Oh, I didn't hear you tell me to say. Okay, I see.
The resolution for this microscope ranges from 1 nanometer to 20 nanometers. The magnification ranges from 20 times to 1,000,000 times, and the max depth of field is up to several centimeters. And on the right is an image of the interior of a scanning electron microscope, which is very similar to the one we have at the ASDRP facility.
All right, so narrowing it down into our project and the use of AI and ML in the field of electron microscopy. So AI can kind of be roughly described as the imitation of human intelligence and into like non-human operations. And one of the best implementations of artificial intelligence is in the operation and automation of routine tasks. And ML is one of the AI methods that is best suited for these applications onto the electron microscope because of the fact that a lot of the electron microscopy, especially with the scanning electron microscope at our lab, is tedious and manual labor that can be well automated. And machine learning in the field of electron microscopy is nothing new, as one of its first uses was almost 25 years ago by Bonnet and his group. So the practical implementations had to wait for more computational tools and resources available that we could work with large scale and large dimensional data sets. But some of the basics are how machine learning can learn from past experiences to pretty much optimize the settings of our SEM to get the highest quality and highest accuracy images. Machine learning can also help recognize patterns and features that can be used for image analysis, image recognition, and can also increase overall efficiency of the microscopy process. And finally, it can adjust imaging parameters such as beam intensity, focus, and magnification. And without getting too much into the depths of electron microscopy, these are pretty much the settings and parameters that manipulate how the image is taken and the features of the image itself. So machine learning being able to identify the best parameters and the best inputs for these parameters would really be able to increase efficiency, quality, and accuracy. So focusing in on the deep learning aspect of the machine learning in electron microscopy. So a lot of the deep learning in SEM automation uses neural networks, which teaches the computers to process information, data, and overall just quote unquote think like a human does. And it can be the process of a neural network can be shown as like a graph with interconnected nodes that resemble how information flows inside a human brain. And deep learning in the image recognition part of electron microscopy is trained to label and kind of analyze each pixel. And it takes a analysis of the nanoparticle images, which are the small details, and it pretty much can focus in on each pixel and label it to being part of an overall picture of the feature, saying like curvature, alignment, or shape, which shows how each pixel is part of a greater feature within the entire image. And an example of deep learning is the denoising of an image, which is when the model can quote unquote remove the noise or the excess details from a pixel, which can make the picture a lot more usable and increase the efficiency of it being analyzed. And it can be used in computer vision. So motivation and goals for this project include that the automation using machine learning of this SEM operation can save time for the routine users, like researchers and quality control technicians who have to do the same thing over and over again to make sure that the quality and results of their SEM experimentation are accurate. And improving image consistency is also a big, really important aspect because we don't want different operators to produce different results, which increases the precision or, in scientific terms, the reliability of our analyses and our results. And it is very well known that the loading and unloading of samples into the SEM is lots of time consuming and multi-step routines that a computer can perform pretty well. And tweaking electron optics for high quality imaging is a must, but yet again, very tedious, repetitive, and human labor. So, and lastly, examining the same basic samples repeatedly can be boring, tedious, similar to the other two uh, two actions. And again, the AI, machine learning, and just a computational version could really like speed things up.
So now I'm going to talk about SEM experimentation. So when a human operator does the experimentation, they have to go through the steps of tuning and positioning. They have to select a region of interest in the image. They have to adjust the focus, magnification, and astigmatism, and they have to choose any detectors. So these are all tedious processes. And instead of us doing these routine operations, we can have our artificial intelligence and machine learning do this. And an example of this, which is seen in Kalini's paper, is a pre-trained machine learning network that can be used to identify an object or feature of interest. And based on that observation, they can go through a predefined sequence of action. And next, this is what our AMRay SEM looks like.
During the semester, we have made individual flowcharts of the SEM SOP, which is the standard operating procedure, that we merged together. The master flowchart has been sectioned off into 4 main swim lanes: the startup, the loading of the sample, the image or picture taking, and finally the cleanup. Next, we broke up our master flowchart into more detailed flowcharts. First is the flowchart of getting started with the SEM. The first swim lane describes mounting the sample as you want to pick a suitable sample. The top left photo is ASDRP's current choices regarding sample holders. The next swim lane describes the process of loading the sample into the SEM. During this time, it is critical to check that the pump and vacuum controls are correct. The bottom left image is the pump and vacuum controls of the SEM machine at ASDRP. Finally, the last swim lane depicts preparing for imaging. In this step, it is critical that you slowly turn the filament knob because that is where the SEM is most vulnerable to be damaged.
Much more isolated. Her story continues after the latest BBC.
The next flowchart describes the imaging process.
BBC News with Stuart McIntosh. Fierce wildfires in Western Canada are burning close to a number of large towns in the North Alberta and British
The
Columbia.
low magnification image is an example of what the sample might look like. Then you want to switch to TV mode since that will allow you to see the image in a more current time. Once you do that, you want to find your feature of interest, then increase magnification. Focus the image by going in and out of focus. Increase magnification, then focus again. And finally, increase the magnification again and adjust the focus and astigmatism. Once you do that, you want to go to a lower magnification because you always want to focus on a magnification higher than your image to have a clear image. The middle magnification and lower magnification photos focus on different parts of the sample. Once you have the image you want to capture, you want to switch to a lower scan for a cleaner image and then freeze. You will continue to repeat finding features of interest until you have all the images you need. The final flowchart depicts shutting down the SEM. The first swim lane depicts turning off the filament and voltage and depressurizing the chamber. Then the next swim lane depicts removing the sample and repressurizing the chamber. Then finally, the last swim lane depicts detaching the sample from the holder, taking the gloves off, and then finally locking out.
So from this discussion, what we learned is the loading and unloading samples for the SEM and the routine and how they take about 20 to 30 minutes to do. And we also learned how to sample view the images that are in the SEM and how to use them in low, mid, and high magnification and how time consuming they are as they have many steps. And if one step is not followed, the image could be damaged or, it could just not be that good. And that's why we can use AI to optimize this step. So the potential focuses of areas of automation are robotics, and we can create mechanics for loading and unloading the samples. But this is very complex. As you've seen, the SEM, and it's such a big instrument, and you have to make sure things like the temperature is correct, the pressure is correct, the gun valve in it is closed, and all those controls to do that are all around the SEM. So the bot has to be able to travel from one side of the SEM to the other side of the SEM without damaging itself in the SEM and without forgetting any of the steps. So that's a really complex task to do. Next part is the user interface UI improvement and sending signals if anything's wrong to the person controlling the SEM, and the electron beam optics and managing it for the standard reference in sample image recognition. Then we have the sample positioning, where we use AI to detect the image of the sample, and we position the sample in a correct place such that the electron beam can hit it properly, and the secondary electrons that bounce back from the sample can help create a good image. And when we position the sample correctly, we'll get a really good image. And using AI to control that sampling position will really help.
In terms of future plans, so we know that automating tasks like feature recognition and loading and unloading can be pretty challenging. So to keep things realistic and manageable, we're going to focus on one portion of the process for the next couple months. Over the next 3 months, our plan is to collaborate closely with Mr. Downing, who'll guide us with the software part of this. We'll look into coding and research and languages such as Go, Rust, as well as Python. Mostly, we'll be focusing on layer 7 of the OSI, or the Open Systems Interconnection Model, also known as the application layer. This is where we're going to use current applications and technologies to go into our SEM process. This is important as we want to make do of our current automated parts of the machine. That way our focus can be tuned in onto one portion. By the end of our summer, our aim is to replace the current image manager with a more efficient solution. This will not only optimize our workflow, but also free up valuable computer cycles. Additionally, we'll be exploring various machine learning models to find the best fit for our specific needs. This means looking into algorithms and techniques that can help us automate tasks like feature recognitions more effectively.
So the future plans for the image recognition and sample positioning projects. So we want to buy SEM reference samples for low and mid and high magnifications. And we want to start generating data sets of images so that we can train the AI to help detect the samples from the images. And we also want to look into available data sets for the scanning electron microscope. And we want to study and select suitable structured layered algorithms so we can design the machine learning to detect the sample. And that can require a lot of complex machine learning algorithms. And we want to also learn mapping for the low resolution corrupted images onto high resolution. So if an image is corrupted, we want to see how we can use AI to help construct to turn that really low resolution image into a high resolution image with AI. And yeah.
Here's some of the references we used. We mostly based our research on Kalini's paper, but we looked and researched at many more. Thank you, ASDRP, for listening. Do you guys have any questions?